So here's the next question. What happens to the pyruvic acid, or the pyruvate? Well, it depends on what the cellular conditions are like. If there is oxygen present, pyruvic acid will enter the matrix reactions and then the membrane reactions. That's if oxygen is present. If no oxygen is present, or oxygen conditions are very, very low, we'll just say it undergoes fermentation. And that's a whole separate scenario we'll talk about too. Actually, it's called lactic acid fermentation. It's a little different than the fermentation that's used to produce ethanol in wine or beer. But same idea. Okay, let's talk about those matrix reactions then. This would be scenario 1. If oxygen is present, pyruvic acid undergoes oxidation. That's the first step in the membrane reactions. Pyruvic acid or pyruvate oxidation. And then, after the oxidation of pyruvic acid, then the product of that enters into the citric acid cycle, which you may also know as the Krebs cycle. Dr. Krebs was the person that worked out the mechanism for this cycle. But it really, the citric acid cycle is called that because it does involve citric acid. The same acid that's in lemons and oranges and limes and most of your citrus fruit. Or all of your citrus fruit, I should say. Okay, so somewhere you should have that carbohydrate summary table that we filled out last time. I forget what page it is. 23. 23B. $31, I think it is. Yeah, 31. So let's go through and just summarize the matrix reactions here. What's the first thing on the column? Location? Respiration type, okay. Respiration type is aerobic. Don't have oxygen, you can't go forward with this reaction. Location, where does this occur? It occurs in the mitochondrial matrix. If we look at a mitochondria, Remember, mitochondria are the powerhouse of the cell. This is why. We'll get it. All this explanation as to why they're the powerhouse of the cell. So mitochondria has a double plasma membrane, and the inner membrane is infolded like this. These little infoldings create a lot of surface area. They're called cristae. And so what we have is this space inside the inner membrane. It's called the matrix. The mitochondrial matrix. That's where these reactions are going to take place, hence the term the matrix reactions. They're occurring in here. This is the cytosol out here. This is where glycolysis takes place. This is where the pyruvic acid is, the pyruvates. So the pyruvates are going to have to make their way into the matrix. This space in here is called the inner membrane space. So when we talk about the matrix reactions, we're talking about this space in here, inside that inner membrane. We talk about the membrane reactions, we're talking about the reactions that occur across from the matrix to the inner membrane space and back again. Those are the membrane reactions, which we'll get to on Wednesday. All right, reactants. We're starting with pyruvic acid, or pyruvate. NAD. FAD. ADP. Products. Get ATP, NADH, FADH2, oh, and a pretty important one, carbon dioxide. It's these matrix reactions that produce the carbon dioxide that we exhale. All right, so somewhere you should have this diagram. (21B) maybe?
23, sir.
23. So this top part here pretty much is the oxidation of pyruvic acid part. This down here is the citric acid cycle. And it's a cycle because the starting point is also the ending point. So here again, the circles represent carbons. So we start with two molecules with 3 carbons each, and they're going to enter one at a time. So what happens is our first pyruvic acid comes in. It undergoes oxidation. NAD plus is reduced to NADH. So we're picking up some of the electrons and protons from the pyruvic acid. That's what allows NAD to become NADH, is reduced to NADH. We lose a carbon dioxide, and we're left with a 2-carbon molecule called acetic acid. Acetic acid is the acid that's in vinegar. Vinegar is just very, very dilute acetic acid. So we move from a 3-carbon molecule to a 2-carbon molecule. Here is that 3rd carbon in the form of carbon dioxide. We've captured some electrons in the form of NADH here. So approximately how much ATP is this NADH worth? It's a 3 ATP equivalent, we'll see. Now what's going to happen is that 2-carbon acetic acid will be carried into the citric acid cycle. But it can't do it by itself. We need another molecule. This molecule is called coenzyme A, CoA for short. The coenzyme A is what will carry that acetic acid into the citric acid cycle. The coenzyme A does not get consumed in the reaction. It is, in a way, acting as an enzyme. Because it's not being consumed in the reaction, it can get used again. It is an organic molecule. Once it delivers the acetic acid into the citric acid cycle, it detaches and can go back and pick up another acetic acid. So what happens then at that point is that the 2-carbon acetic acid combines with a 4-carbon molecule that's already present called oxaloacetate. 4-carbon molecule. So the 2 carbons from acetic acid, the 4 carbons from oxaloacetate, combine to form a 6-carbon molecule called citric acid. Hence the term the citric acid cycle. Now, if we look closely at the citric acid cycle, what we're going to see is that we eventually go from 6 carbons to 5 carbons to 4 carbons, and eventually we get back to that oxaloacetate again. And that oxaloacetate is reused for another acetic acid molecule. Yeah?
What's the coenzyme A doing? Is that helping it to bind with the oxal?
It's simply picking up the acetic acid and carrying it in and essentially pushing it into the reaction with oxaloacetate. It then releases the acetic acid and goes back up and picks up another acetic acid. Okay, let's go through the citric acid cycle then. Again, all these reactions are facilitated by enzymes. Unless I tell you the name of the enzyme, don't worry about it. So, citric acid is converted into a similar 6-carbon molecule called isocitric acid. It's a little different because what's happened is it's lost a water molecule. Carbon has shifted. So now we have 2 carbons directly. One carbon has 3 other carbons attached to it. That's isocitric acid. Now we're going to see that we go from 5 carbons to 4 carbons. And we also see that NAD is going to get involved. And wherever we see NAD, we're going to have an oxidation and reduction reaction. So NAD is going to be reduced to NADH. We see that we lose a carbon in the form of carbon dioxide. And now that isocitric acid has been oxidized to a 5-carbon molecule called alpha ketoglutaric acid. Yeah. We have another oxidation reduction reaction occurring. Again, we see NADH reduced, or NAD reduced to NADH. We see carbon dioxide released, so the alpha ketoglutaric acid is being oxidized to a 4-carbon molecule. This is always the one I... Give us a name for it or not? I think this is called succinate, but you don't need to know that one. I'll have to double check. But it is a 4-carbon molecule. Now at this point forward, we're going to stay a 4-carbon molecule. In the process of going from 6 carbons to 4 carbons, the NAD has captured electrons and protons. And we know that that energy then is going to be used to make ATP and electron transport. Now at this point, it's just a matter of getting back to the oxaloacetate to run the cycle again. Now what happens here is that a phosphate gets added. And in the process, what we get is an ADP becoming an ATP. So we do get a molecule of ATP forming directly here. Actually, it's indirectly. I've left out a step just to simplify things. But we're still getting ATP. So now we actually end up with a slightly different 4-carbon molecule. And this is where FAD comes in. FAD is going to capture some electrons and protons from this second 4-carbon molecule here. It will become another 4-carbon molecule. Water gets added back in to change the shape of this molecule to this one. So let's see, we can do 4-carbon 1, 4-carbon 2, 4-carbon 3, 4-carbon 4. They all have names, but I think you have enough names at this point. So the 4-carbon number 3 is converted into a 4th form, and then that 4th form undergoes oxidation and reduction. NAD is reduced to NADH. The 4th 4-carbon molecule is then oxidized to oxaloacetate, and we're back to where we started. We're only half done, though. How come? If we start with a single glucose molecule in glycolysis, and we follow it through all the way to the end, what we've just done, we've only done half. How come? There's 2 pyruvic acids. We've only looked at 1. Let's total everything up before we start the second pyruvic acid. So over here on the side, ATP, NADH, FADH2, carbon dioxide. At this point, how much ATP have we produced from the first pyruvic acid? How much ATP directly have we produced? 1. How much NADH have we produced? 4. Don't forget about the oxidation of pyruvic acid. If we're just talking citric acid cycle, yes, it's 3. But if we talk about the matrix reactions, then it's 4. How much FADH2? 1. How much carbon dioxide? 3. That's from 1 pyruvic acid. We have a second pyruvic acid that needs to undergo this entire process as well, which would give us 1 more ATP, 4 more NADHs, 1 more FADH2, and 3 more carbon dioxides. So what's our total carbon dioxide we've produced? 6. Doesn't that fit with the 6 in the original cellular respiration reaction we looked at? Yes, it does. So for every single glucose molecule that starts in glycolysis, goes through glycolysis, those 2 pyruvic acids have the potential to go through the matrix reactions and yield a total of 2 ATP, 8 NADHs, 2 FADH2s, and 6 carbon dioxides. So you got this chart again, right? Go ahead and dig that chart out. So let's summarize and total everything up here in the matrix reactions. Did we use ATP in the matrix reactions? No. We didn't use ATP, but did we produce ATP? Per glucose. What's our potential yield of ATP in the matrix reactions? 2. Per pyruvic acid, it's 1, but per glucose, it's 2, because for every glucose, remember, we get 2 pyruvic acids. So our net ATP produced directly in the matrix reactions is 2, right? How much NADH did we get from the matrix reactions? Per glucose, 8. How much FADH2 did we get per glucose? 2. Now remember, we said that there's a total of 36 to 38 ATP produced in cellular respiration. Well, 4 of those are right here. 4 of those are produced directly. How many NADHs do we have? 10. Each of those NADHs has enough energy to produce at the maximum 3 ATPs, right? So how many ATP is that? 30. Now, each FADH2 has the potential to yield a maximum of 2 ATP each when they get sent to the electron transport chain. So what's our yield of potential ATP from the FADH2? We have 2 produced with a potential of 2 ATP each, 4. And if you add those numbers up, what's the potential for ATP? 38 ATP. That's where the 38 comes from. The 36 is another story. I'll get to that Wednesday. Yeah, it should be FADH2. Should be FADH2 on your table. It's a typo. So what we'll see then in the membrane reactions is that these 10 electron carriers will give up their electrons and their protons, and that energy then will be used to make ATP in a very specific way. I'm going to do it on time here. I know it's messy. But these diagrams are on Blackboard, and you can download those. I put a few links on Blackboard for you that are a couple animations. I found this, I don't know if anybody have an iPad or an iPhone, but there's an app called the Khan Academy. It's free, and this guy was a stockbroker, and he made lots of money. He's like 35 years old, and now he just makes videos all day on various aspects of science, and he's got a bunch on there on this stuff. So there's an app for free, but I think he's got a website too. You can go right to the website and watch stuff right from the website. It's called the Khan Academy, Khan. Just Google the Khan Academy. And I'm pretty sure it's Kahn or K, yeah, something like that. Here. Khan Academy. He's got stuff on biology, chemistry, organic chemistry, healthcare and medicine, physics, cosmology and astronomy. If you click on the biology, he's got all kinds of stuff on there. Stuff on ATP, introduction to cellular respiration, oxidation reduction review, oxidation reduction cellular respiration. He's got glycolysis, citric acid cycle, electron transport, all the stuff we're going to talk about. Okay? All right, take a few minutes and keep in mind and think about the fact that every single pyruvic acid needs to go through this. And it's important to look at this from the perspective of an entire glucose molecule. Let me show you this. So we have one glucose that becomes two. We have one glucose that becomes two pyruvic acids. Each of those pyruvic acids need to feed into that citric acid cycle. You need to be familiar with what happens and the numbers that we get from each one of these. The whole purpose of these reactions is to slowly peel away a little bit of the energy contained within these molecules so that it can be used to make ATP. If we stopped after glycolysis, we'd only have a net of 2 ATP. But by going through these other reactions, it's much more efficient as far as energy transfer is concerned. There's much more energy being utilized In fact, this may sound like a low number to you, but aerobic respiration is about 40% efficient. We're actually capturing about 40% of the energy from that original glucose molecule. And that's what becomes ATP. 40%. What happens to the other 60%? Remember I said the law of conservation of matter? Matter can't be created or destroyed. Energy is the same way. Energy can't be created or destroyed. It can be transferred from one form to another, from one molecule to another. But you can't make, the sun is the only thing that can make energy from scratch. 40% is not very efficient from our perspective. Would you buy a furnace for your house that's only 40% efficient? No, it may as well be burning dollar bills. Where does that other 60% go? Somewhere else. Where? It's lost as heat. Heat. When these bonds break, heat is released. That heat is important. It actually helps to maintain your proper body temperature so your metabolic reactions can occur. Why does your body temperature go up when you exercise? Because you have a lot more metabolic reactions, a lot more ATP synthesis occurring. And so how does your body compensate for that extra heat? So to keep your temperature, you sweat to help you cool down. All right, so on Wednesday we will continue with the membrane reactions. I will give you a little short practice quiz on Wednesday over this stuff. It will not be counted as your grade. It'll be just answer some questions that I ask you. Okay?